Thank you for standing by and welcome to the Imugene Investor Webinar, focusing on Monday's announcement of positive early data from the Vaccinia MAST study. Joining us from Imugene today, we have the CEO and Managing Director, Leslie Chong, as well as the Chief Medical Officer, Dr. Paul Woodard. All participants are in a listen-only mode. There will be an initial slide presentation lasting for approximately 15 minutes and we'll take questions thereafter. If you'd like to submit a live question, please do so using the Q&A panel within Zoom. We've already received quite a few on email and we'll get through as many as we possibly can. So to kick it off, I'll hand it over to Leslie. Please go ahead.
Thank you, Matt. I am delighted to review our recent exciting news. So I'll just go right into it. Disclaimer. So those of you who have been with Imugene understand what we're doing, but our value proposition or for our investors' interests, we have an advanced portfolio with multiple shots on goal. We have leaders in development of allogeneic CAR T cell therapy, as well as in solid tumors. We have, along with Dr. Paul Woodard and I, we have an experienced management team with over 150 years in combined experience, not only in drug development, but also in drug approvals. We have ongoing clinical trials in diverse solid tumors, as well as hematologic or blood cancers. And we have a robust cash runway, but you've heard me say this before, I use it pretty prudently because we really want to protect and conserve our cash resources. We have 4 unique platforms, but in which one we're going to focus on today is the CF33 oncolytic virus, especially our Vaccinia study and our MAST study. Look at those happy faces. So Dr. Paul Woodard and I have known each other for more than 18 years. Actually, when I met him, he had a 1-year-old and now she is an adult, which is so incredible to believe. So since we've met, we've gone on to several different companies, specifically in biotech companies. So we've got lots of experience and we have developed drugs together. So I am going to let Paul take it away.
Very good. So I'm going to review CF33, our Vaccinia program. We could go back just one slide. So CF33 can infect and selectively kill cancer cells. So if we follow along in this cartoon, we see a virus. The virus then enters the cancer cell, it duplicates and reproduces, and then it ruptures the cell and releases, not only kills the tumor cells, but releases thousands of new virus particles, which can affect other cancer cells in the area. We've done some engineering enhancements so that the CF33 only infects and kills cancer cells. It is possible to engineer it to carry payloads to increase tumor cell killing. And it kills tumor cells in multiple different ways. It can directly kill tumor cells, as we've shown in the cartoon, but it can also activate immune cells to kill cancer cells. And as these tumor cells die, portions of the cells can actually be presented to the immune system, which is called priming the tumor environment to enhance the patient's immune response. And there is precedent for approval of oncolytic viruses with 3 different oncolytic viruses approved in different regions. Next slide. This is the study design for our phase 1 MAST study. So on the left-hand side, we see Vaccinia or CF33, the monotherapy dose escalation, and there are 2 parallel cohorts, intratumoral or IT, in which the virus is directly injected into 1 or more tumor lesions, and intravenous. And we started at a dose of 1 × 10⁶ and are planning on continuing to escalate. We're currently at the 1 × 10⁸ dose level in the intratumoral cohort and the 3 × 10⁸ dose level in the IV cohort. We also have a combination part of the study with pembrolizumab, the anti-PD-1 molecule. And intratumorally, we're in the middle of dose escalation at the 3 × 10⁷ dose level, and intravenously, we're at the 1 × 10⁸ dose level. Once we're settled on the appropriate dose, we will pursue expansion cohorts and one or more different tumor types of interest. Intratumoral will occur first. Next slide. So this study, MAST, stands for Metastatic Advanced Solid Tumors. We've treated 34 heavily pretreated patients to date. The doses have been safe and tolerable thus far, and we have 25 patients who are currently evaluable for response. That means they've received drug and they've had their first scan at 6 weeks of treatment. And with that, we've seen activity already. We've seen a complete response in a patient with bile duct cancer and another partial response in a patient with melanoma and 16 patients with stable disease. This is the monotherapy data. We also have a combination patient with bile duct cancer who's had stable disease for going on 5 months now. There have been 7 patients with gastrointestinal cancers who've received CF33 alone. This includes patients with colorectal cancer, bile duct cancer, pancreatic and liver cancer. And 6 of the 7 patients had disease control, which gives us a disease control rate, meaning either a complete or partial response or stable disease of 86%. We do plan to expand in at least 10 additional patients with a bile duct cancer, and there's the potential to pursue other solid tumor types at a later time. This study is being conducted at 12 centers in the US and Australia, and some of the sites are shown on the right-hand part of the slide. Next slide. So this is what we're trying to do. We're trying to turn cold tumors hot. So when you have a tumor, some tumor types are what are called hot tumors in which they have immune cells present. Those are typically more responsive to checkpoint inhibitors such as anti-PD-1. But most tumors are immunologically cold, which means they don't have immune cells in the tumor or around the tumor. And so this shows the patient with a complete response in bile duct cancer, which is a cold tumor type. If we start on the left-hand part of the slide, we can see the baseline CT scan, which is at the start of the study before the patient has received any drug. And what we see is an abdominal lesion, which is shown in the crosshairs with the red oval around it. Interestingly, we followed this patient at their second scan, which was after 12 weeks of treatment. We observed a phenomenon called pseudoprogression, and that looks like the tumor has increased in size. And indeed, the measurements showed that it had, but what you can see is the tumor has hollowed out into a cyst, which is the dark portion of the tumor that's shown. By the third scan, this tumor had started to shrink further. And by the fourth scan, the patient had a complete remission, and all that's left is a cyst present. And this is a patient who had received 3 prior lines of different chemotherapies. They were PD-L1 negative, which is a poor prognostic sign, and they'd had no response to any of their therapies. And then they come on CF33 and have had a CR, which has been maintained for quite a long period. Next slide. And so one of the tumor types we would like to expand into are bile duct cancers. This is also called cholangiocarcinoma, and we're specifically looking at patients with unresectable or tumors that can't be completely removed by surgery. These patients are usually treated with chemotherapy. We've got shown here the first and second line chemotherapy. Most patients also now receive checkpoint inhibitors in the first line setting. About a third or less of patients have one of a variety of different kinds of mutations, which you can target with specific antagonists, and some of those are shown in the middle portion of the slide. And then finally, rare patients with bile duct cancers have high tumor mutational burden or have high microsatellite instability, and that means they have higher numbers of mutations and are potentially targetable by checkpoint inhibitors. Next slide. I will turn it back over to Leslie. Thank you.
So, Paul, I just want to mention how bile duct cancers generally do not respond to a lot of these different therapies, although they are, well, it's not a curable kind of tumor. So, can you speak a little bit about that?
Yes. So patients, there have been a couple of recent large phase 3 studies in first-line cholangiocarcinoma patients, and the median survival is about a year with the best current therapy. Most patients unfortunately relapse, and the response rates to therapies in the second or third or late lines of treatment are very low, and the survival is very low once patients have relapsed. So it's really an unmet need, and we think it's an area that we can continue to investigate and hopefully end up with an approval at some point.
Thank you. So I just want to share with you our very exciting value inflection points for the next coming up 12 months. As you can see, we have lots of activities with Azer-cel, our allogeneic CAR T program in blood cancers. On Carlytics, of course, we've had our first patient in, and we have a lot of opportunities here to do combinations, as well as announce on several different combos opening. When we announce the IT combo expansion or the intravenous combo expansion for onCARlytics, know that we have reached a nice dose, and that's why we're exploring. Much like the vaccinia MAST study, we're dose escalating because we have seen meaningful safety, low tolerability. So we want to go higher in the dose to explore further opportunities in several different tumor types, as well as further dose. We are quite excited to announce that we will be opening up that expansion of 10 additional patients in bile duct cancer to start at one of the doses that we're currently interrogating. So lots of activities ongoing for the next 12 months. So I'm pretty excited about several different things that it is ongoing in Imugene. So that's the end of our presentation. So we'd love to entertain some of your questions.
Thanks, Leslie, and to you too, Paul. As you've mentioned, we'll move on to the Q&A session. Again, if you'd like to submit a live question, please do so using the Q&A panel within Zoom. The first question I have is, will there be an additional indication besides bile duct cancer that will be a part of the MAST expansion study?
Paul, do you want
to
answer that?
Yes, I would expect so. So we're early on in development still, and we'd be looking for other tumor types to potentially expand into.
Thank you. The next question is there any chance that vaccinia would be trialled against mesothelioma? Human Fong was at a conference in Tasmania and demonstrated vaccinia's killing power against this disease. Given this disease is almost always fatal, might this be a compelling case for accelerated approval from the FDA?
I'll take that. So we actually have treated a patient with a variant of mesothelioma called testicular mesothelioma. And that patient did have some tumor shrinkage in some of the injected lesions. But mesothelioma is a tumor type that could be potentially enrolled in this study or our onCARlytics study.
Thank you. When can we expect to see data from the vaccinia combination arm with Keytruda?
That's also relatively early on in development. We're at the second cohort. So it's as that data evolves, we'll plan on presenting that at a medical conference.
Thanks, Paul. As an investor and patient, will you be trialling for metastatic breast cancer or HER2 breast cancer?
That's another tumor type that would be eligible for either the MAST study or our onCARlytics study.
The next question is, what's the new timeline for the MAST trial? It was scheduled to end in 2 years, but with an expanded doses and additional indications, has this been revised? And related question, will the bile duct expansion be done in dose escalation or only once optimal biological dose is found?
Do you want me to take that, Paul?
Sure. So we've seen very good safety and we're already seeing clinical activity. I think we want to continue to determine the best, safest, and most active dose before we expand into specific tumor types. You're going to get a better understanding of how the drug works if you do it that way.
Thank you. What's the timeline or steps for applying for breakthrough therapy?
I would stay tuned.
No problem. The next question is, will MAST be finished prior to additional vaccinia trials being started?
I think that question is about potentially a indication and another phase 2, perhaps, I think. Once we finish out some of the expansion, and if we see clear signals, then we could easily turn that into a expedited phase 2, but that remains to be seen. We want to follow the science.
Thanks, Leslie. The next question is, there is no planned mono/combination plan for IV vaccinia currently. In the November presentation, it appears to mean intratumoral only.
That data of the bile duct as well as the others were in monotherapy, which is great because that lends itself to combination. We're at the lower doses, the mid dose for the combination. So once that study, we look at the data, and if we want to submit to major cancer conferences that Dr. Woodard was talking about, then we will provide that data. However, it's in the mid dose, and it's at the lower dose of our vaccinia.
Thank you. The next question is, why are you starting with IT dosing for the vaccinia expansion study?
Well, because that's where we've seen opportunity. So if we can actually get some good signals from the monotherapy, that road to registrational study or to market study or furthering the studies is much easier with the monotherapy.
Thank you. Crosses over a little bit with a previous question, but is the aim of the bile duct trial expansion to receive an orphan drug status and accelerated approval?
So we're seeking further signals with that, but I would stay tuned.
Thank you. The next question I have, bear with me. Have the patients in question received subsequent therapies?
Oh, yes. So they are all well treated. Some lines of therapy, we have a patient that's on his 6th line, meaning they've taken 6 different therapeutic opportunities and have progressed or relapsed or failed off those, and they ended up in a phase 1 study such as ours. Yes, so they're well treated.
Thank you. The next question is, how do you perform the intratumoral administration for cholangiocarcinoma? Do the patients need a laparoscopy, and is it done via ERCP, assuming it is not accessible?
You can, it depends on where the metastases are. So if you have a skin lesion that's easily targetable, a lot of these patients have liver metastases. Those can be targeted with various approaches. You can do it through a needle approach, or I suppose you could do it through an ERCP or other interventional radiology type approaches.
Thank you. The next question is, I was hoping to see ovarian and pancreatic cancer patients benefit from the MAST study. Will you be recruiting these patients in future?
Those patients can be included in the study and the oncolytic study as well. As the data sort of evolves and we have more patients with those tumor types, we will present that data.
Well, actually, the oncolytics, our first patient is ovarian.
Ovarian, yeah.
Yeah.
Thank you. The next question I have is, do you expect data to be better with patients who haven't had prior treatments and therefore a potential better immune system?
Well, the FDA is it's
Go ahead.
ethically so doctors or oncologists cannot give you an investigational product if there is a standard of care. So fortunately, unfortunately, we have an opportunity to affect tumors when it's that late stage. So the fact that we have a bile duct cancer complete response, that's, again, as Dr. Woodard explained about cold tumors, there's no reason why that patient would have responded to our therapy other than our virus did in fact replicate and draw the immune system there. So that means there is a mechanism of action that's working. So we're quite excited actually to be in late line of therapy and still affect these kind of changes at such an earlier stage.
Thank you. Not sure how much we can add to this, but have you seen responses from patients dosed intravenously?
We're seeing early signals. We're encouraged, but we're at a lower dose. So that's one of the reasons why we're dose escalating. So Dr. Woodard and I feel pretty strongly that once we get into a higher dose of IV, we'll see much more of an effect.
Thank you. I know this was touched on during the presentation, but does chemotherapy work for bile duct cancers or just delay progression?
There is no cure. Yeah, there is no cure. I think if you read all the research and reports, chemotherapy just potentially staves off growth for only a short period of time. So there is no cure for bile duct, and chemo just seems to subside it for a small amounts. There's only the survival rate for bile duct cancer 5-year rate is anywhere between 2% to 24%. And that 24% is if you've had 100% resection or surgical removal at an early diagnosis. So this is clearly an unmet need. And, we're pretty excited that we've seen 2 patients thus far respond to our therapy, and especially in the mono, as well as some IV. So we've got a speckling of various different solid tumors. The fact that we could triangulate and find one that is working looks like very efficiently. And then the fact that we're expanding, we'll likely find more as we dose escalate.
Thank you. The next question is there any idea on the mode of action and what the difference between the complete response and those that had stable disease is?
Well, the mode of action is what we showed in the slide. So it can directly kill tumor cells. It can recruit immune cells. And then as you sort of chew up the tumor cells, your body processes all these proteins from the tumor cells. And the immune system can recognize that. So you're helping all of that with CF33 or vaccinia. I think you're also asking about, do we have translational research data yet? And we don't, but we will publicly show that when we have more data.
Thank you. Next question is, what do you think about the value being created here for the company?
To me, data is value for the company. That's exactly what we're here to do. So Dr. Woodard and I want to affect the oncology or cancer landscape and provide better therapeutics for patients, and patients are our North Star. So whenever we can produce data and great outcomes for this, for this bile duct cancer patient, I think about him being able to go home and tell his friends and family that he is without any tumor every time he gets a scan. So I think the data is our value, and we are building more and more value as we collect more data.
Thank you. The next question is, what sort of side effects have been seen at the highest dose?
Well, that we've seen some level of fatigue, all those things that you would encounter almost like flu-like symptoms, but they're all manageable and they go away, more importantly.
Thank you. This is a follow-on from a few questions ago, but how long do standard of care rules take to change?
That's a good question. So it generally takes a phase 2 or 3 study to change treatment patterns that shows a benefit to survival or regression-free survival.
Thank you. The next question is, did the imaging of the responder patient with bile duct cancer show presence of the virus in the tumour?
Oh, they're talking about the HNES and then the SPECT scan. Yes, there were bioreplication, and we did see CF33 in the tumor.
Thank you. And just one final one I've got is, how large is the bile duct cancer market versus in comparison to other markets?
Well, we don't really look at the market size because when you can affect a later line, then if you then become that standard of care, that product or that therapeutic can move into other lines. So you have to look at it from that perspective.
Great. Well, thanks once again to you, Leslie, and to Paul. That's all we've got time for today, and concludes the Imugene Investor Webinar and Q&A. There will be a replay of this available shortly after this live session concludes, and anyone can catch up on it there. But thanks again and to everyone for attending today, and we look forward to speaking to you again soon.